Hi, welcome to the cerebellar seminars. Today, as our guest, we have Michael Mauk, who is a professor at Department of Neuroscience at University of Texas at Austin. Mike received his PhD from Stanford University, where he was a student of Dick Thompson, one of the leading figures in cerebellar research. Mike discovered that one could use the inferior olive as the source of the unconditioned stimulus during the study of eye blink conditioning. He then did his postdoctoral training at Stanford with Jeff Kosis, and then joined the faculty at University of Texas at Houston, then moved to UT Austin. Mike has mentored some of the leading figures in the field of cerebellar research, including Javier Medina. Mike's work combines experiments and computer simulations to investigate the neural basis of learning in the cerebellum. All yours, Mike.
Thanks, Reza. Thanks for the invitation. Thanks for everybody to be here. I'm going to take the hat off, but yeah, sorry about that. Okay. So let me introduce the people who helped me with this. So you see there on the right, Lydia and Thibaut have been working on the 5 inputs project about.
I don't have the sound. Yeah.
Oh no, you're unmuted.
Somebody needs to mute there, sorry. Lydia and Thibaut's a colleague of mine, a theoretical colleague. We've been working on why granule cells have so few inputs. Satvik and Sean have been working on the forgetting project that I'm going to talk about. JD's collaborating with Hiroshi Nishiyama, another colleague of mine, on some in vitro LTD work that's testing predictions from our work. And then Carter's a new graduate student who is working on several of the projects that I'll list at the end as ongoing. So many of you may know that we've been studying the cerebellum for a long time using eyelid conditioning and combining experiments with computer simulations of the cerebellum. And so let me just start with a little preview about how that all fits together before I get into it. So this is these are eyelid conditioning raw data. So what you're seeing in each of these traces is eyelid position as a function of time in a rabbit. So up means the rabbit is closing its eyelid. Eyelid conditioning is a simple form of associative learning where in the old days we would pair a tone with an air puff or some sort of electrical stimulation of the eye. In the same way that Pavlov would have done bell and meat powder. This is the first day of training for this rabbit, so you can see it doing is reflexively closing its eye to the stimulation of the eye. We portray the duration of the conditioned stimulus, the tone, in black here to help you see when the responses are. This is a typical first day. We don't see a ton of learning in the first day. This is also a typical second session, same rabbit now 24 hours later. And you can see that early on, large and robust conditioned responses. This is the rabbit closing its eye in response to the tone and not to the stimulation of the eye. We care about this learning because it comes from the cerebellum. And so I'm not going to show you a whole lot, but it's a surprisingly rich behavior. It's bidirectional. The rabbits can learn and then unlearn or extinguish. There's a timing component that I'm going to show you in a bit. There's savings where relearning is faster than original learning. So it's a surprisingly rich behavior, but the reason we care about it is because it comes from the cerebellum. And so this is really the key to everything that we do, is that it was starting in Dick Thompson's lab and moving on from there. The series of discoveries in the 80s and 90s showed that when we play a tone in one of these experiments, it activates mossy fibers that come from the pons and provide a mossy fiber input to the cerebellum. When we puff the eye or stimulate the eye, it activates inferior olive neurons in the eye face region of the sensory map there, and that provides a climbing fiber input to the cerebellum. So mossy fiber, climbing fiber. The learning happens with plasticity on the cerebellar cortex and the cerebellar deep nuclei. The deep nuclei, then there's a map of the body. So there's sort of an eyelid region of the deep nuclei, the interpositus nucleus. Those neurons project to the red nucleus, and then the red nucleus down to, we don't see the facial nucleus here, but down to the premotor and motor neurons that make the eyes closed. And so what's important here is on the output side, this output pathway is pretty fixed. If you stimulate in a naive animal in the right spot here, you get full-blown eyelid responses, and those responses don't change with training. And we often use electrical stimulation of mossy fibers as a replacement for the tone. My thesis project in Dick's lab a zillion years ago was showing that you could stimulate this region of the, you could stimulate the climbing fibers electrically as a replacement for the threat to the eye. So really the key part about eyelid conditioning is that it lets us control inputs and measure the outputs of the cerebellum. And this is really important. So this is sort of how I think about eyelid conditioning is for our experiments, it lets us control the inputs to the cerebellum. And when we measure those eyelid responses, we can infer quantitatively, because we've done recordings, we can infer quantitatively what the output neurons in the deep cerebellar nuclei are doing. So one corollary of this is that most of the behavioral properties or eyelid conditioning are a reflection of the input-output processing properties of the cerebellum, which is what we care about. That's the main focus of our work. Now, it's also true then that as we've used simulations to study the cerebellum, it really helps us test the simulations in a biologically relevant and quantitative way, because we can give the simulations mossy fiber and climbing fiber inputs that we know occur in our experiments in rabbits, and we know what kind of output under most circumstances, in previously tested circumstances, that the simulation can do. So we can quantitatively measure when the simulation is succeeding and failing. And so all of that together that has let us use experiments to control inputs and measure outputs, use simulations that we can test in really relevant ways to ask what's going on in the cerebellum, what are the computational properties happening in the cerebellum, what's the purpose of every component of the circuit. This has been what we've been up to. Now I have to say, oh, by the way, this is our latest, greatest picture of our latest, greatest simulation computer. This is about almost 100 teraflops of computing power in one little box sitting in the lab. So the amazing advances in parallel processing and computer power have enabled much of what we're up to. And I will say that I think everybody should consider building simulations of what they're trying to study, but to be prepared for the reality that they will disappoint. So for the longest time, we would build these simulations and they would fail in a variety of ways. And that's extremely important part of our approach is we don't tweak them to make them work. If they fail, we try to use that as information. And there are a number of papers that we published over the decades where the idea came from grappling with failures. And so in the beginning, they failed a lot. And then in the middle, they failed certain ways and succeeded in other ways. And we've always tried to use both of those to help inform what we were up to. But in the end, I have to say that in the last couple of years, the simulations essentially can replicate everything that eyelid conditioning shows us that the cerebellum does. And so I'm going to talk about one way that we can make use of that. So this is one of the behavioral properties of eyelid conditioning. This is the timing. So these are real eyelid responses where we've varied how long the tone is on. In other words, how much the mossy fiber input precedes the climbing fiber input in the end. And you can see that the cerebellum can learn over a wide range of those, ranging from 1/8 of a second to almost 2 seconds, 1.5 seconds here. This is in milliseconds. And you can see that the timing of those responses and the variability of the timing of those responses is influenced by that so-called interstimulus interval. This was the aspect of cerebellar learning that was the hardest nut to crack, and that we've only accomplished in just the last few years, thanks in large part to the discovery by Wade Regehr's lab that Golgi cells inhibit each other. And so we're in this interesting situation now where finally, after 32 years, the simulation can quantitatively replicate everything that we see about the cerebellum from eyelid conditioning. And so I'm going to tell you just the basics of how learning works to set up what we're going to be working on. So ignoring a lot of the detail in real cartoon form here, then here's kind of the main learning pathway in the cerebellum. That I have CS here for conditioned stimulus, but sensory input and motor commands activate mossy fibers, which then diverge onto that very large layer of granule cells. Granule cells make excitatory synapses onto Purkinje cells, which inhibit the output cells, and eyelid responses come from when these deep cerebellar nucleus neurons fire. Learning happens because climbing fibers control the induction of plasticity at these synapses. When these synapses are active more or less in the presence of a climbing fiber input, they undergo LTD. And when they're active without that, they undergo LTP. And so the kind of the gist of learning then is that if we pair a tone that activates mossy fibers, I've cartooned 4 of them here as being activated by 4 granule cells as being activated by my tone. And then LTD is induced at those tone activated synapses onto the Purkinje cell. The Purkinje cells then learn a pause in their ongoing activity, which disinhibits these cells, and along with some plasticity in the deep nucleus, produces the burst of deep cerebellar nucleus neuron activity that makes the responses. This is actually a peristimulus histogram from a real Purkinje cell. And just to show how much that learning at the Purkinje cells is important and how much it controls everything, here's a recording of a Purkinje cell during just a typical eyelid session. And so this is a raster plot of the Purkinje activity where each row is a trial and each dot is a spike. And the conditioned stimulus time is shown by this gray rectangle. And you can see that this is a Purkinje cell that it's the same one from the previous cartoon that pauses to help make these conditional responses. But to show just how tight the coupling is between the Purkinje cell activity and the behavior, we have the computer sort the conditioned responses according to their latency to onset. So you can see here's a few where there were non-responses, and you can see these are the latest all the way up to the earliest onset responses. We have the computer sort the raster plot in the same way and then draw a red dot where it detected the latency to onset of the response. And you can see that on a trial-by-trial basis, these Purkinje cells pause about 40 or so milliseconds before the eyelid starts to move. So when there's no response, the Purkinje cell didn't pause. When the Purkinje cell pauses late, you get a late onset response. When the Purkinje cell pauses earlier, the response begins earlier. So there's a lot going on, but for what we're going to talk about today, we're really thinking about how do the Purkinje cells learn these pauses, and what are the ingredients that make that happen. And just to show you then that this is a smooth firing rate of virtual Purkinje cells trained at different interstimulus intervals. And if you remember from the previous slide, the rabbits could learn when the time between the mossy fiber input and the climbing fiber input was 1500 milliseconds. And so you can see that the simulation Purkinje cells can learn the same thing. These are in effect Purkinje cell conditioned responses here. These are the Purkinje cells pausing. We draw this 80% line here because we know from empirical recordings that for the typical rabbit, when a Purkinje cell response falls to 80% of its ongoing level, that's when the eyelid moves. So these demark our estimate of when the latency to onset of the virtual eyelid responses would be, is when it. And you can see now that, and this is the thing that we couldn't do for the longest time, is that the simulation can learn over the same range of timing intervals that the rabbits can, and it can show the same kind of timing of the condition responses that the rabbit. So this is my take-home message, that finally, after 32 years, the simulation can do most of what the real cerebellum can do. So what that lets us do then is we can kind of have fun, whether this is a simulation inside of this box or thinking about the real circuitry. It allows us to kind of pivot to this new kind of project that, frankly, I didn't anticipate until things started to be the way they are now. That we could use the simulation, which works and it captures all of the key properties of eyelid conditioning, to say, well, why does the cerebellum have the properties that it has? And so this is what I want to talk about today, and which is why I chose the audacious title that I did, which is why the cerebellum is built the way that it is. So I'm going to talk about, we have a lot of projects like that. I've chosen 2 of them because I think I can squeeze them into 40 minutes. One of them is studying the fact that the simulation forgets. The simulation forgets despite the fact that it has perfect synaptic weights, and by perfect I mean that there's no imperfection in their ability to hold on to the strength that they have. There's no fading or turnover of proteins that make that happen. And I want to say that this work is very much inspired by a paper from Larry Abbott's lab in 2005 that talked about how forgetting can occur just because of the way activity is happening at night between sessions and such. Then I'm going to talk about briefly about work that we've been doing with this classic interesting question of why do the cerebellar granule cells have so few excitatory inputs from mossy fibers. So again, this is the Fusi, Drew, and Abbott paper that we really drew some of our inspiration from, and I love these 2 sentences where they talk about how the main challenge in thinking about long-lasting memory is protecting the memory trace from the ravages of ongoing activity. In other words, plasticity is happening between our training sessions. So it's protecting it from ongoing activity and not just from the ravages of time. And evidence suggests that forgetting can be a consequence of ongoing activity and not merely just the passive decay of plasticity from imperfect molecular mechanisms. And so the fact that the simulation mimics eyelid conditioning so well, and the fact that it forgets seemed like a fun test bed to help think about the ideas that Larry and his people came up with. And I wanted to start with talking about this. So just to sort of set up the way that we would do these things, this is all simulation work now. So on the left, I'm showing the way that we might train the simulation. And so this is a raster plot from one of the virtual mossy fibers. Excuse me, all of the mossy fibers are assigned a background activity, but a small percentage of them are told that when we play the tone or the conditioned stimulus, that they increase their activity to around 100 hertz. And so this is a raster plot of one of these CS activated mossy fibers. And so we would train the simulation in this way by activating them and then having an excitatory conductance onto the climbing fibers in the simulation. And the simulation learns. So this plot's virtual eyelid response. And so the simulation learns these virtual eyelid responses that look for all the world like real rabbit eyelid responses. And you can see it takes about 100 trials for it to learn. So that's learning. And you'll notice the x-axis here is trials. Well, then we can just mimic time, pass it between sessions, the rabbit sitting in its cage. We just run the simulation for hours and hours with just background activity. And now you'll notice that the y-axis is still the amplitude of these virtual learned eyelid responses, but now the x-axis is just time. And you can see over the course of about a virtual day, the simulation can forget what it learned. And we set out to quantify rigorously that this forgetting comes from random walk of synaptic weights driven by ongoing activity. And there was a surprise along the way that I want to show you about. So this project really highlights part of what's fun about doing simulations. Is that you can measure anything, you can control almost anything. And so here what we've done is we've trained the simulation like I showed, and then we can download the synaptic weights of the Granule to Purkinje synapses. There's 1.2 million of them, 2 to the 20th of them. And here's a frequency distribution of those 1.2 million synaptic weights. Most of them are around 0.5, which is near where they start. But after training and the simulation, having learned responses, there's some number of them that have gotten weak. They've undergone LTD. And you know that these are the guys that are driven by the conditioned stimulus and whose changes have permitted the acquisition of these conditioned responses. But we really wanted to focus on these and in terms of forgetting. And so I'm going to show you some things that we did to show that these are the guys doing the heavy lifting in terms of the simulation learning these virtual responses. But I want to pause right here and just remind you what the hypothesis. The hypothesis is that between sessions with the occasional plasticity events that happen from the background activity, sometimes LTD, sometimes LTP, that there's a diffusion. I just want to remind you what random walk looks like. So if we just start a bunch of synaptic weights at 0.5 and we just randomly increase them and decrease them and with the same sizes that we do in the simulation, you can see the expectation is that they should all stay at 0.5. But the idea behind a random walk is that even though the expectation they will, as the number of steps grows larger, they spread out, that the reliability of that expectation decreases. And you all know that the simple form of that random walk equation is that the standard deviation of this is equal to the square root of the number of steps. Flip a coin 1,000,000 times and your ratio to heads and tails is going to spread out according to the square root of n. Now I just wanted to show that ours is a more of a bounded random walk because most of them the weights are starting at a boundary, that they're at near 0. And but the math is similar and the process is the same, as that you just let them run and random walk says that they spread out. Now, it was Einstein who proved that diffusion is from random walk of molecules bouncing around, and so we can say that then the hypothesis here is that forgetting in the simulation is this random walk driven diffusion of synaptic weights spreading out and not remaining reliably where they're supposed to be. Okay, so back to the weights then. So we wanted to figure out just how many of these weights should we care about, and so we did an experiment where after training we would pick some number of them and return them back to their original weight. We would reset them back to their weight, and we would do this from weakest to strongest. And so the x-axis here is the number that have been reset, and the y-axis is this virtual eyelid response. And you can see that as we reset them, we can make the conditioned responses go away. And we have to reset about 20,000 of these guys to make the conditioned responses go away. So that gives us a sense of about how many weights we need to care about in terms of if those guys change, then there's going to be problems with retaining the response. A variant of that then is rather than resetting them, we could after we've trained the simulation, we could just freeze some of them and say, you're not allowed to change anymore, and then run forgetting and see if that stops forgetting. And we did that, but also freezing more and more of them, systematically adding them from weakest to strongest post-training. And so when we did that, so here's our normal learning curve where we don't freeze any of them, or learning. Forgetting curve, this is the forgetting over the 1 day of, and here we are, here's the forgetting when we freeze the weakest 10,020. And so bottom line here is we need to freeze about 30 or 40,000 of those weights to prevent forgetting. So if we freeze those weights, then the simulation becomes impervious to forgetting over time. And it's kind of interesting. It's interesting that you have to freeze a few more of them to stop forgetting, then you have to reverse them to make a response go away. And we've had lots of fun discussions about why that is, but it is sort of the same range. And so now the next manipulation was supposed to be the coup de grace, and was really going to show that forgetting is about random walk. And so what we can do then now is instead of reversing them or freezing them, we can start with this simulation that's been learned. We can take some number of these weights, and we can download them from the simulation and use a little Python program to implement some number of steps in a random walk, and plug them back in and ask, well, how much have you forgotten? And the hypothesis is that if random walk driven diffusion is forgetting, then if we walk them artificially, we should see the same level of forgetting. And here's where the wheels fell off a little bit. So here again in black is the normal forgetting of the simulation. Blue here is when we did this artificial random walk for 30,000 of the, so really almost no forgetting. So we thought, okay, we didn't do enough of them. So just to cut to the chase, we did that fake random walk on all 1.2 million of them, and still a huge discrepancy. So this was a surprise, and it took us a while, I have to say. But what we came to understand is that there was more at play here. It was still a random walk. But what we had to come to grips with was this, is that the real standard deviation equation for a random walk includes both square root of n and this variance term. So if you're flipping coins, where there's an equal probability of heads and tails, and the size of a head is the same as the size of the tail, that variance term is 1, and you can ignore it. But in our circumstances, for example, we already had to incorporate this in because it's just the nature of how the cerebellum works, that LTP events are about 9-fold more common than LTD events, which means for equilibrium sake, the size of LTD steps has to be 9 times bigger so that all of that balances out in equilibrium. That was all factored in to our random walk, and yet it still wasn't working. So we did this and that. We made sure that it wasn't drift, that the synapses were drifting rather than walking. And ultimately, what we finally came to understand is that the discrepancy was coming from the fact that granule cells tend to fire in bursts. And firing in bursts, and remember, it's when they fire that they're eligible to undergo plasticity. And so the fact that they fire in bursts means that they're eligible for plasticity in bursts, and that kind of violates the independence of this. And what it basically, for the purposes of this equation, means is that you tend to have a smaller number of larger steps. So a burst might entail several LTD or LTP events, which would all be usually in the same direction. So again, it was the equivalent of changing this equation where there were a smaller number of steps of larger size, and when we incorporated that in, when we incorporated the burstiness of granule cells into our fake random walk, then it matched. And so that was a bit of a saga, I have to say, but it was fun to figure that out. So the summary of that little project then is that we asked whether the ongoing plasticity as imagined by Larry Abbott's lab could do forgetting. And the idea here is that a random walk of the weights causes a diffusion of synaptic weights, which causes a forgetting. And the answer was yes, but we had to factor in the fact that the burstiness of the granule cells has a large effect on this process. And so we were really doing this as a foundation for testing hypotheses about things in the cerebellum that might resist this forgetting. And here, these three things are literally just the three specific aims on a forgetting grant that I wrote. These are things about the cerebellum that might slow that forgetting process. But we already discovered, even in this, so-called foundational experiment, that the burstiness of granule cells has a big effect. So it's surprising what we don't know about plasticity. And if you build simulations, you realize that our understanding of plasticity is much more incomplete than most people think. So one thing, we know that when the granule cells are active, they become eligible for plasticity. And the way we implement that is their eligibility for some amount of time. It's that some amount of time that makes this problem with the random walk. So one possibility then is maybe the molecular mechanism of that eligibility means that during this burst, it's sort of eligible once. It's one eligibility event rather than a range over time, and that would alleviate this. So anyway, this is the kind of thing that we simulation people look back at the LTD, LTP people and say, hey, can somebody, here's something that nobody maybe thought about that we should be thinking. The other thing is, I just want to say, because there have been some drift in the other direction about this, here's another reason to believe that granule cell activity needs to be sparse. The more background activity of granule cells there is, the more steps there are in the random walk and the faster forgetting. We've built simulations where granule cells were far less sparse than normal, and the simulations forget really fast. So just a little throw in there. So that's forgetting. Let me completely shift gears now and give you a very brief version of another project that we've been doing. And I have to say, this started as a simulation project, and we discovered that we could get plenty done with just math. So this is going to be a little mathier and a little less about the simulations for now. I'll also say that there's a lot of math and a lot of components to it. And for the purposes of this talk, I've challenged myself to distill it down to the two most important ideas that come out of this work. So this is more multifaceted than I'm going to present, but I'm going to try to distill it down to a really clear, pithy take-home message about why granule cells have to have a small number of inputs. So here's Ramoni Cajal's drawing of granule cells, and they have these little claws for dendrites, and the synapses of mossy fibers only happen on those claws. And so we know that the average granule cell has 5 of those, so it gets input from 5 mossy fibers. And one of the most amazing little things ever, we know that those are 5 different mossy fibers, because mossy fiber presynaptic processes are these rosettes. And the spacing of these rosettes on a mossy fiber is wider than the span of the typical thing. So we know that it's very rare for the same mossy fiber to provide more than one input to a granule cell. So it's a very intriguing computational question. Half of the neurons in your brain are granule cells. That's one crazy thing about them. The other one is what's the computational point of them having only 5 inputs from the mossy fibers? I want to acknowledge that we're not the first people to think about this, obviously. So Mauk talked about it. He kind of portrayed it as granule cells are, fussy little feature detectors. And then Larry's lab did this beautiful paper where they had this interesting metric, which was coding dimensions per input, which peaked at 5 to 7. And I find that intriguing, but I have no idea what that means. And so, and then Angus Silver's group did this really mathy thing where they talked about lossless coding and mutual information. And I'm aware that what I'm about to tell you may be some way related to what these guys have said, but if nothing else, we're really searching for something that's a little more intuitive, something that I can understand. But I just want to point out that somebody better at math than me might point out that what we're saying has a fair amount of overlap with what's already been done, but I'm going to go for it anyway. So here's what we're thinking about. Of course, we've got, when you have a fixed number of mossy fibers diverging onto making synapses onto a larger fixed number of granule cells. And so here's some number of mossy fibers converging onto a granule cell. Mossy fibers then have to diverge and talk to some number of granule cells. And again, our question is why is this convergence so small? Why is it 5-ish? One of the things I want to point out is that if there's a fixed number of mossy fibers and a fixed number of granule cells, then we should keep in mind some of these truths, which is the number of granule cells times their convergence has to equal the number of mossy fibers times their divergence. That just means that there's the same number of presynaptic elements as there are postsynaptic elements. This leads to other things. These are important, and they're part of why what I'm going to show you is true, and we could come back to this, but I just want to point out to you that when we think about changing the convergence of mossy fibers onto granule cells, we have to remember that we're also changing the divergence of how many granule cells each mossy fiber talks to. Okay, so here's my attempt at the really intuitive explanation of this. If we think about a granule cell that has input from 5 mossy fibers, I've labeled them 1 through 5. And we imagine that it has a threshold of about 3. In other words, 3 of these 5 have to be active. We did a whole bunch of math. It was kind of complicated to make things really gentle. And then I had to laugh when all of the terms started to cancel and this, and I literally laughed out loud when I realized that the answer is that under that circumstances, if we're thinking about one of the mossy fibers, each mossy fiber, given this granule cell of convergence 5, threshold 3 is active, each of the mossy fibers has a 6 in 10 chance of being active. And I've just kind of showed you this here. Here are the combinations of 3 that include 1, and here are the combinations of 3 that don't, right? And this is true over a wide range of convergences and thresholds. This is just true. That if a granule cell is active, then the probability that each of its mossy fibers might be active is a threshold divided by convergence. So you can see, for example, that a granule cell of convergence 5 and threshold 5 very reliably signals, this is not rocket science, that mossy fibers 1 through 5 are active. Okay. So let's explore the implications of this, and I'm going to show you results from just a little simple simulation where we have some number of mossy fibers, and we choose about 100 of them to be active, and we're going to look at we're going to figure out what we can learn from the activity of each of the active mossy fiber, or some number of active mossy fibers, and we're going to accumulate how much certainty we have that each mossy fiber might be active. And so my graphs look like this. So this is a simulation where the granule cells have convergence 5 and threshold 5, and we looked at about 128 of them. And mossy fibers 1 through 100 are the active ones, and so we can accumulate through joint probabilities the expectation. Yeah, we're really certain that those 100 are active, and we're really certain this is 100 of the several thousand mossy fibers that weren't active, and we're really certain that those guys aren't. So it just takes a couple hundred or so mossy, look at it, 100 and so granule cells to infer as sort of an ideal observer that these mossy fibers were active and these mossy fibers weren't. If we drop the threshold to 4, Now, it's still pretty good at telling us, you look at about 128 granule cells again that are active. We can tell that, it's still pretty good. Our average certainty of that the active mossy fibers are active is about 0.99. But you'll notice now we've got some misinformation that some of the granule cells are not active. We're kind of suspicious that they are, because remember, there's an in each of these granule cells that are active with threshold 4, there's an imposter who isn't necessarily active. These are the imposters. Right? So let's do some more comparisons. Here's that same granule cell, convergence 5, threshold 4. It's good at saying which granule cells are mossy fibers are active. It's got some misinformation. But if we compare a granule cell with convergence 25, and I chose threshold 11 because that would produce about the same amount of granule cell activity for any given mossy fiber input. You can see again, pretty good at telling which mossy fiber are active, but the misinformation has now grown rapidly. The average estimate here, the average likelihood that we think one of the active granule cells is active is appropriately at 0.9. Here, the average likelihood of the non-active granule cells is about 0.94. It's risen to almost, we're about half certain that most of these mossy fibers might be active too. And this is because there are a lot more, for every active granule cell, there are 11 mossy fibers that are really active and that are 14 imposters. And in fact, when threshold is below half, there's more imposter misinformation than there is information. And you can say, look, I don't care about the way that you equated. You could equate the other way, which is now we can do compare convergence 5, threshold 4 with convergence 25, threshold 20. That's threshold is 80% of convergence in both cases. Still a lot more misinformation. Again, because there are 5 imposters here. There are 5 mossy fibers that may not be active, even though this granule cell is active. There's only 1 here. So the interim summary then is that when a granule cell is active, the probability that each of its constituent mossy fiber inputs is active is threshold over convergence. And that's just a simple mathematical fact. When granule cell, so what that means is where granule cells where T is near C provide the most information, actually they provide the less inaccurate information about which mossy fibers are active. Good granule cells, that if you wanted to build a layer of good granule cells, you would want their thresholds to all be near their convergence. Now here's the thing. If we switch gears and think about, well, given those parameters, how many granule cells could be active? It's kind of a simple math to show. So this is the equation that determines what proportions of granule cells would have T number of inputs given their convergence. So C is convergence, T is how many inputs are active, would be active, and M is the proportion of mossy fibers are active. And so this equation can tell you how many, if you want to know if you have convergence 5 and some amount of mossy fiber activity, how many of the granule cells are going to have T of their inputs active, as the active ones. And you can produce curves like this where here's now, based on that equation, this is for convergence 5. So the x-axis is the proportion of mossy fibers that are active. And the y-axis is the proportion of granule cells that we would expect to be active for if their threshold was 2 inputs, 3 mossy fiber, 4 or 5. And you can see this makes sense when all the granules, all the mossy fibers are active, all the granule cells have enough inputs, right? We can also sort of remember that we can probably ignore the right half of this, that we don't really think that more than half of the mossy fibers are active in any given time. And we can also remember that granule cell activity is pretty sparse. So we're probably looking here that, if about 25% of the mossy fibers are active, we're going to want a threshold of about 4 to have, I don't know, 1% of the granule cells active. So here's the same result for convergence 25, and here are the thresholds from 2 to 25. And what you can see then is that for sensible high thresholds, there isn't much activity. So if we want 25% of the mossy fibers active and we want about 1% of the granule cells, we'd have to have a threshold of 11. If we wanted that threshold 20, because we want threshold to be near convergence, that's a granule cell that's never going to be active. It's just a combinational argument that granule cell is almost never going to be active. Here's an example, just picked from those curves. If we imagine there's about 23% of the mossy fibers are active, for a granule cell with convergence 5 and threshold 4, about 1% of them are going to have 4 or more of their mossy fiber inputs active. For convergence 25 and threshold 20, one granule cell in a billion would have 20 of their inputs active. So, I mean, sure, granule cell activity is sparse, but the rabbit cerebellum has fewer than a billion granule cells. Our cerebellums have 40 billion. And so if this is the way you built granule cells, about 40 of your granule cells would be active at any given time. So the idea here is that because of the way threshold grows slowly, so this is taken from those curves, and we're picking some fixed amount of mossy fiber activity, and we're shooting for a fixed amount of granule activity. This is the threshold that you would need to produce that. And you can see that as convergence grows, threshold grows very slowly with a slope under 1/3. So what that means is that if you have a like I've shown, if you have a granule cell with convergence 25 or 50, a sensible threshold is going to be 11 or 14 or 15. You can't build granule cells with high convergences and have their threshold near the convergence. So this is the summary of that. And this is our main argument for why granule cells have to have a small number of inputs. Granule cells with a threshold that are significantly smaller than convergence are bad. They provide a lot of misinformation. They will tell you, they will imply to you that mossy fibers that aren't on might be on. Thus, a good granule cell needs its threshold to be near convergence. The thing is, though, is that as C grows, the threshold required to produce a sensible amount of granule activity grows slowly, so that with granule cells with convergences greater than, say, 10 or so would almost never be active given the amount of mossy fiber activity sensible that we can imagine. So in simple terms, then, a good granule cell with a high C would never fire, with a high convergence would never fire, or you can't make a good granule cell with a high convergence. So you just combine these two things. Good granule cells need their threshold to be near convergence, and you can only do that when convergence is low. So anyway, those are two examples where we are using the fact. Now I get it, that was mostly math. There's some simulation work there, but to present it briefly, I skipped most of our simulation ideas about that. So what I want to emphasize then is that if it's interesting to realize that once you have a simulation that actually mimics the cerebellum, that you can start to ask why the cerebellum the way it is. This is a list of the kinds of things that we've been working on. Why inferior olive neurons are electrically coupled, why they have subthreshold oscillations. Are there homeostatic plasticity processes that we could plug in there that makes the cerebellum self-tuning, like the real cerebellum is? Why do you need granule cells? Why do they have 5 inputs? What functions do the cerebellar collaterals that loop, the deep nucleus collaterals that loop back around and become mossy fibers? So we can start asking questions about why the cerebellum has. And I gave you 2 simple examples here, but I wanted you to sort of walk away thinking about the kinds of doors that are open when you start to understand a system well enough that you can play with why the way it is. So again, Lydia and Thibaut did the work on granule cell inputs. Satvik and Sean are the forgetting people. JD and Carter are lab members doing cool things that I didn't talk about today. So thanks much.
Thank you so much, Mike. And if anybody has a question, please raise your hand. I will call on you. Mike, if I may ask you about the first part of the talk, you focused on the plasticity of the granule cell Purkinje cell synapse. In your model, in order to produce the pause in the Purkinje cells, I assume there is a role to play for the molecular layer interneurons. I wonder if you could talk about that.
Oh man, so they're really important. And in fact, what's fun then is that in part through experiments that help clarify things, we can appreciate that, for the longest time, we just lumped stellate and basket cells or molecular layer interneurons are just, they get input from granule cells and they inhibit Purkinje cells, fine. Maybe you need two types, whatever. Well, in the simulation, I can tell you that basket cells and stellate cells subserve entirely different purposes. That the basket cells help synchronize Purkinje cells so that they kind of speak with one voice when they talk to the deep nucleus. That came out of some work that was collaboration with George Augustine's lab. We've done more simulation work since then than we showed in that 2018 paper. The stellate cells were pursuing this interesting idea that the reason why it's cool that they inhibit the dendrites of Purkinje cells is they help control the induction of plasticity and make it more sensible and a little more stingy. So the idea there then is that there's been some empirical work to suggest that the dogma that a climbing fiber affects all synapses onto the Purkinje cell the same may not be true. It may be controlled by local inhibition. And so both of them play important roles. One we've worked out pretty well. The other one we're working on. One of the things on my list back there was the role of stellate cells in helping control the induction of plasticity.
Thank you so much, Mike, and thank you for the link to Synchrony. Jennifer, you have your hand up.
Hey, Mike, really nice to catch up with you and what you've been doing. I agree with Sasha. You look exactly like when you were on my thesis committee, which was a very long time ago.
She said when you erased the bulb, but that's okay.
I wanted to ask where you're getting the assumption of LTP being 9 times more frequent than LTD, and what other assumptions that depends on in the model, like how sparse the granule cell firing is or whether they fire in bursts. And if I can have 2 questions, the other question is in the model, what percentage of your granule cells fire in bursts versus other non-bursty patterns?
Most of the granule cells fire in a bursty way. I mean,
and burst, what does burst mean? Like less than 100 milliseconds or?
5 to 8 spikes at 1 to 300 hertz. The plasticity part is completely independent of anything about granule cells. The ratio of plasticity events. So one of the things that the simulations helped us understand the best, I think, is that the role of the 1 hertz spontaneous climbing fiber activity. And so it's regulated to a level, because it's regulated by cerebellar output, it's regulated to the level that the total amount of LTP is balanced with the total amount of LTD, right? So if the LTD window is 100 milliseconds, and the math is easy because I'm assuming that the spontaneous equilibrium level is 1 hertz. That makes the arithmetic easy here. If the LTD window is 1 hertz, then the LTP window is 100 milliseconds. The LTD window is 900 milliseconds. That makes LTP events and spontaneous during background 9 times more frequent than LTD events. Now, the only way that you the only way that the simulation can have a 1 hertz, given that, the only way that the simulation can have a 1 hertz spontaneous climbing fiber rate and can regulate to that is that the LTD events, which are less frequent, are 9 times larger, that the synapse change are 9 times larger for an LTD event than they are LTP. In fact, it's really amazing, but all physiology aside, all circuitry aside, the thing that controls the spontaneous level, that spontaneous equilibrium level of climbing fiber activity in the simulation is the ratio of the size of the LTP and LTD events. If you don't make them 9, and your LTD window is 100 milliseconds, then the spontaneous level will drift to a different value than 1 hertz.
Thank you, Jan. Thank you so much. Solomon, go ahead.
Thanks, Mike. 2 quick questions, if I may. One is, do you know if during the eye blink conditioning, now that the eye is closing, perhaps the input to the mossy fiber is changing too, because there are now motor commands driving eye closure. Do you know if or anything changes in the mossy fibers as the animal learns, or is all the changes that you observe is in the Purkinje cell?
No, the mossy, Abby's work with Indira, with the mossy fiber collateral, most of the deep nucleus neurons have a collateral that go back to the cortex and turn into a mossy fiber when they get there. So 5%-ish of mossy fibers are driven by cerebellar output, and that output changes during learning, so the mossy fiber changes. And
okay, and now a quick one, if I may. In terms of this forgetting due to the random walk, these evidence that have been emerging that in eye blink conditioning, even after learning, the complex spike firing is not fully gone. Perhaps actually it shifts. Javier Medina shows that it shifts to be pre air puff or pre-eye closure. Do you think those may have a role in kind of preventing this forgetting due to random?
Well, I don't know. We don't see that in rabbit, right? So I don't know if that's a mouse thing or rabbits are weird, but there's nothing in the simulation that makes that happen.
I see.
And I have to say that, it's my understanding that mouse eyelid conditioning, they have trouble getting extinction. And I think they do because there's something about their CS that activates climbing fibers. And so I don't know if that's a mouse thing or, like I said, rabbits are weird, but
I see.
If I'm just thinking about rabbits, that doesn't happen. And the simulation doesn't do that and doesn't need that.
Cool, thanks.
Thank you. Sasha, please go ahead.
Mike, this is beautiful. I'm not the only person on the better late than never schedule, I guess. Like, this is really cool that you actually got the model to work. My question, and it's just great to see you. So my question is, how much of your conclusions about the cerebellum as a whole are actually interpositus and fast spiking interpositus thing, as opposed to, for example, a small cell dentate or fastigial thing?
Well,
so a different way of asking the question is to say, this is sort of like the question I thought Jennifer Raymond might be asking, which is, how do your conclusions vary as a function of the circuits that you're studying?
Well, I wrote a paper a long time ago about simulations and reminded people that simulations are always incomplete. And you can think of those as errors of omission, that you just don't have everything in there. We just try to avoid errors of commission, which is adding things to make it work that aren't real. Our deep nucleus is primitive by your standards. We have 2 types. We have the excitatory ones that are the output, and we have the inhibitory ones that loop back to the inferior olive. Now, there are things that happen in the virtual deep nucleus of the simulation that are important for its behavior. The timing, it has an influence on timing. The idea about savings is almost entirely from the deep nucleus. But I think that if you looked at representation, our representation of the deep nucleus, you would rightly conclude that there's some work to be done there. There are errors of omission.
Fair enough. I'm actually wondering more about the cerebellar cortical differences across dynamic and structural differences across.
Yeah, I mean, in my primitive world, the cerebellar cortex is all the same. And I think I agree with the idea that each of them could, each part of cortex could tune itself given the particular statistics of the inputs or outputs that it has to deal with. But, you know, I'm a one-trick pony in that way. We think about the part of the rabbit cerebellum that deals with eyelid conditioning. So.
Thank you, Sasha. David, please go ahead.
Hey, Mike, that was really fun. And congratulations on, you know, your 32-year quest reaching this point. So I have a couple of questions, one about the mossy fibers and one about the parallel fibers. The one about the mossy fibers is your conclusion about the optimality of the 5 inputs dependent on some assumption about the statistics of how the mossy fibers are active? Say, if I could rephrase that, so if a granule cell receives one mossy fiber that's CS activated, is it more likely to be receiving other mossy fibers that are also CS activated? Or are each one of those things a flip of the coin? And if so, how does that correlate with what we know physiologically about the encoding?
That's a great question. So in the simulation itself, the wiring of the mossy fibers to the granule cells would slightly increase the likelihood that a granule cell might receive more than one input from a CS activated mossy fiber. But the math that goes into the little arguments that I presented are independent of such things. It really only doesn't care whether one of them is from background and one of them is from the CS. We're really just asking if in general my threshold is X, how likely is that, you know, given this mossy fiber input, that X will happen.
But if there is much more convergence, how does the behavior of the model change? In other words, let's just posit right now that you know, that these mossy fibers tend to come in bunches.
Right.
You know, the probability of a particular granule cell, if it's got one CS input, is that it's much more higher that it's going to have others than of another randomly picked granule cell.
Right.
Does that change things in the way it plays out?
Do you think that would be true for random background activity?
Well, no, I think it might be true for CS evoked activity.
Well, then that could increase the amount of activity that those, you know, convergence 25 neurons could get. But we would still have to grapple with the fact. So it's weird. So you would think that background activity kind of shuffles all that.
Huh.
And these things need to be true for both background and like the inputs, the learning inputs that we care about. So, but I'll grant you that if you're thinking about a you know, a targeted like CS-like input, that would provide an opportunity for any given granule cell to have a higher than expected number of active inputs.
All right, can I ask a parallel fiber question too?
Sure.
Yeah, so right, so you've got a weight of the parallel fiber to Purkinje cell synapse that's a single number. And we know that you know, physiologically that weight is a combination of pre and postsynaptic processes, and we know that there's LTP and LTD on both sides of the synapse. And so, like, does that matter? In other words, I guess, you know, how reasonable is it to model the weight as a single number and assume that LTD and LTP can always reverse each other?
Well, we don't do that. So I didn't
Okay.
talk about it, but the weight is two numbers. There's a long-term weight that can only change according to what the climbing fibers are saying. And then based on some work we did a long time ago, and based on Christian Hansel's work, there's a presynaptic short-term form of LTP that happens when they're active but fades with time.
Mhm.
And so it's two numbers, but I could rephrase your question as, is it valid? I don't know. I don't know. You'd have to tell me whether it matters, whether if there's sort of a presynaptic and postsynaptic contribution to the weight, if I need to break those apart to understand the synaptic conductance. You'd have to tell me whether that's true.
Yeah, well, we'll do that offline.
Awesome. Awesome.
Yeah.
That was wonderful. Well, thank you, everyone. Mike, that was just lovely. And thank you for all the great questions, everyone.
Thank you. It was wonderful to see everybody's faces.
Same.
Great to see you, Mike. Well done.
You. Take care.